Hi guys, my name is Ernest and this is Ernest Sciences and Mathematics TV channel. So in this specific discussion, we're going to look at the concept of a synapse. And basically the synapse refers to the region or the small space that is in between the axon of one neuron and the dendrite of another. So in this specific section, we're going to have a dendrite given as such. And this is going to be the axon of a neuron that is going to consist of these various parts for the synaptic vesicle and other ones like the mitochondria that we're seeing at this specific region. Now, there are chemicals which are going to be found in between this specific junction that we refer to as a synapse, and these are going to be referred to as the neurotransmitters. The neurotransmitters are going to allow impulses to be started in the second neuron, as we're going to see later in this specific discussion. And of course, I'm going to provide another elaboration or rather another display of this specific synapse or this specific gap or the activity that's going to be carried out in this specific region to ensure there's a transmission of impulse. Now, in this specific section, we're going to see that we're going to involve the use of neurotransmitters, which are going to start impulses in the second neuron or the effector muscle or gland, as we're going to see later. Now, in this specific discussion, we're going to see that these are going to be the synapses, or rather, they're going to be the dendrites of another neuron, which of course are going to be involved together with this specific axon. And of course, this has been given in a more elaborate way, which we're going to see in this specific section. So in my next display, I'm going to show the pathway that is going to be followed in the development of this specific neurotransmission, which of course is going to be an activity that is going to lead to, of course, an impulse in the second neuron, as we have mentioned in this specific video. Now, in this specific display, it's going to give a detailed form of a neurosynapse and the mechanisms of this impulse transmission, which is going to be realized in between this axon and the dendrite, this specific end of another neuron. So in this case, we're going to look at it. Of course, it's detailed in some way, but I'm just going to mention it solely so that we can be able to follow it, perhaps in our other discussion in case we come across these specific names being mentioned. So in this particular case, we're going to have the action potential that invades the presynaptic terminal at this specific point. And then therefore, we're going to have the transmitter, which is of course synthesized and then stored in this particular synaptic vesicle. And then we're going to see later that the depolarization of this presynaptic terminal causes the opening of the voltage-gated calcium channel. And as we can see from this specific point, we are going to have this particular channel illustrated as that. So there's going to be an influx of the calcium ions through this specific channel. And of course, the presence of the calcium ion is going to cause the vesicle to fuse with the presynaptic membrane, as we are going to see later being displayed at this specific case. And they're going to be a release of the transmitter into the synaptic cleft via the process that we refer to as exocytosis. And of course, this is going to be transmitter molecules which are going to be released in this specific order. So if you try to look at this section of the dendrite, we're going to have some currents which are going to be released in this specific part. But as we continue with the process, then therefore we're going to see that the transmitter is going to bind to the receptor molecules in the postsynaptic membrane, this specific case. And of course, this is going to be leading to opening and closing of the postsynaptic channels, as we had seen in this specific case. So if you can try to follow this specific, there's going to be release of some ions, and then we're going to have this specific vesicle going to this particular part for another cycle. Now, in this specific part, we're going to see that there's going to be postsynaptic current that's going to cause excitatory or inhibitory postsynaptic potential that is going to change the excitability of the postsynaptic cell. Now, in our study of course, the concept of impulses, I said that the excitability is basically the ability of a neuron to respond to stimuli. And this is going to be involved in converting it into a nerve impulse. So therefore, we are going to see that the retrieval of this specific membrane is going to occur in this particular case. And the cycle is going to continue in that specific order in order to continue with the transmission of impulse. So basically, that's a summary form of the display or this specific explanation for the release of an impulse insofar as this space in between the axon and the dendrite concerned. So catch us in our next video and kindly check our description boxes for various links of other videos which are related to this. So that is it for now. Bye. God bless you. Catch us in our next video.